that would mean the material from starting with electric potential and voltage through current and resistance, resistor circuits, capacitor circuits, and today's lecture. I think what I'm going to do right now is next week, we will have a lecture Monday and Wednesday, and that will be, that material won't be on the exam, but it will be on the final. And then for Friday, what I will do is give you a take-home exam. That will obviously be open book, open notes, open slides, whatever. I'll probably, I'll put it out Thursday evening, and then it will be due Friday evening. So it'll be like a 24-hour thing. And I'm going to put 4 problems on it. So 1 will be conceptual and 3 will be mathematical. So it'll be instead of having the 4 problems for the conceptual section, I'll just have 1 conceptual problem that you explain and talk through, and it'll be graded with partial credit like the other 3 are. So plan for that next Thursday evening to Friday evening. Does that make sense to everyone? Any questions on that? Yeah, so test corrections. What I'm going to do, since we have, it's really kind of hard to hand corrections back for the second exam, where I still kind of have the hard copies of the exam sitting here in my office. For those, I'm just going to give a few points back on the second exam. For the first exam, if you handed in corrections, I'll give you a few points back on the first exam. And with this exam, since it will be all be online, you'll be able to do corrections online, resubmit corrections and hand in online. So we could do corrections via Canvas. So there will be corrections. I'm just going to handle them for the different exams separately, though. Probably, I think corrections for the first exam will get you a 3-point boost on that exam, which will be a third of a letter grade. I'll give like a 3-point curve on the second exam, and similarly on the third and fourth. Next question. So is there a lab next week? There will be. It'll be on RC circuits, I think, at least right now, that's the plan. However, it shouldn't take you all that long. So I plan to give that on Monday. And it will be due probably Thursday. It'll be a good review for material that could be on the exam anyway. But it won't be very long. The exam will be submitted on Canvas, so it'll be an assignment on Canvas. In the assignment, you'll have the exam form, so which you can then download. If you are able to, you can print it out and then put in your answers underneath. If you can't, if you don't have a printer available to you, you could just write your answers on loose leaf or notebook paper and photograph them and submit the photos, kind of like you would submit your report for lab. And the corrections will be done the same way. Any other questions or concerns about that? Again, obviously, I'm kind of going with the flow here and seeing how this works. But hopefully you'll, hopefully we'll make this work, and that you don't expect the exam to be, the problems will be generally what you would expect on other exams in terms of difficulty. The only difference is I'll only give 1 conceptual problem that'll have multiple parts and partial credit instead of 4 separate ones. There will be a crock-mall problem. I'm not sure what he's going to be doing yet because he's quarantined, but he'll be there. So we good? If you want to email me questions later or any concerns you have later, feel free to, or come talk in office hours. So, but just plan for that. Plan for a short lab to be given Monday. I'm still trying to put that together, but it shouldn't take too long, and it'll be a review for the exam. And then the exam available Thursday evening, due Friday evening. So it'll be a 24-hour take-home open book exam. All right. If there aren't any questions, then let's start talking about our physics for the day. So hopefully you guys got the email. I sent you this it was a fairly dull video in terms of its narration about nerve cells and this kind of review of the defining characteristics of a nerve cell. And the reason I sent that is because today, in our last lecture on electrical current and circuit properties, we want to take all the things we've learned over the past week or so and put them together to kind of discuss how signals propagate down neurons. That's kind of our big kind of biological problem that we want to apply the physics we've learned. So this kind of, along with the human ear and the human eye, this is the third big biology example we're going to do. So summarizing that video, what we're going to look at in this class is we want to discuss and really understand how electrical signals move down neurons. In particular, we want to focus on this thing called a motor neuron, which a diagram from the book is pictured here. So a motor neuron is a cell in the body where you have this, and the cell body over here, and then they have this long tail called an axon, which is essentially like the wire in the nerve. It takes an electrical signal from the body and transmits it to another neuron, and et cetera, and eventually it gets down to a muscle. When the signal gets there, the muscle contracts. So some of the properties of this neuron that we want to understand is, one, how are these electrical signals generated? What are they made of? Why are they designed like that? And in particular, the question that our physics of circuits can really help us answer is, how does that signal propagate down this thing called the axon? What determines its speed? What determines, whether the signal gets there uncorrupted or is messed up in some way? All the stuff that we've learned can be applied to understand why the axon is designed or built the way it is, and why the nerve signal propagates the way it does. So that's our goal for today. This false color electron microscope image here is a image of a set of, these are all axons that are propagating, a whole bundle of axons that form what we would call a nerve fiber. So it's obviously false color. It's an electron microscope image. So some review of what we've already kind of learned about the electrical properties of the cell. So one, we have this bilipid cell membrane layer. Might have all these proteins and channels embedded in it, but basically we know it acts as a kind of capacitor because we have charged ions that accumulate on both surfaces. So it effectively acts like two separated plates of charge. The outside tends to have more positive charge than the inside. So the outside is at a higher potential energy than the inside. Really, of course, the cell is usually circular, spherical, or if we're looking at like a cut of our axon in our nerve cell, it's cylindrical. But the main idea of that, it still acts like the capacitor plates don't need to be flat. They could be circular, they could be cylindrical. Capacitors can come in all different shapes and sizes, as long as we have two different separated plates of charge. The positive charge and the negative charge being separated on the different sides of the membrane creates this electric field that we calculated a few weeks ago. And it takes energy to create that electric field, which is why there ends up being a potential energy difference from one side of the cell to the other, from outside to inside. And normally, everything being equal, the outside, where we have more positive charge accumulated on the membrane, is at a higher potential energy than the inside. And in terms of, if we speak of potential energy in terms of voltage, that means the inside of the cell is on average or more or less around 70 millivolts lower than the outside. We set our zero of potential energy out on the outside of the cell. So the outside is at 0 volts and the inside is at minus 70 millivolts. And if the cell's in equilibrium and happy and doing nothing, just kind of hanging out there, this is what we call the resting potential energy, or resting potential for short, of a cell. A motor neuron encodes an electrical signal by changing this resting potential. It moves around charge so that the potential energy stored in this electric field is different. And that difference in potential energy is what encodes our nerve signal. So if we look at kind of a little cartoon picture of our nerve axon here, so we have our cell body is this big circle here. You get my laser pointer. So here, this is our cell body. And our axon is this long strip. The kind of, yellowish or, pinkish parts on the outside, that's our bilipid cell membrane layer. And then the blue part here is the cytoplasm or whatever that's inside the cell. So if we look up at a close-up view of the axon, what we'll have is we have all these ion channels that allow ions to move back and forth that are usually closed. When the cell body gets an electrical signal, what happens is that the ion channels that are really close to the cell body, that signal will cause them to open. So essentially what that signal does is, if the cell body gets an electrical signal from a nearby nerve cell or from the brain, that will cause a slight, a very small change in the potential energy. Only about 15 or 20 millivolts compared. So it will raise the inside of the cell from minus 70 millivolts up to around minus 55. And that is enough to cause these ion channels in the axon, or at least at the close part of the axon, to open. And they allow all the ions to flow in and out such that it completely reverses the charge distribution. So right at this point in the axon, the voltage jumps from minus 70 millivolts up to a positive potential energy of around plus 40 millivolts. So this part of the axon, the inside becomes positively charged, the outside becomes negatively charged, reversing the direction of our electric field and reversing the direction of the change in our potential energy. So this, compared to our resting potential, so the rest of the axon is at our resting potential of minus 70 millivolts. This change where the inside of the cell becomes positive is called an action potential, since this encodes our nerve signal rather than just the cell sitting there doing nothing. And so this becomes our nerve signal, and through a variety of methods, the action potential has to move down this axon to eventually get to the next nerve cell to start this process over again, which will then transmit it a bit further, which will eventually take it to a muscle, which will cause the muscle to contract. So how this works, this action potential, as you can see, it's all based on the fact the potential energy changes because the charges move from inside to outside of the cell. They move kind of in the vertical direction here to switch the charge distribution and thus the potential energy. However, we need to get this to move down the axon. And this is where the different properties of the axon and how it's built come into play. So around the outside of the axon, we have these things, these little blocks of things called myelin, which act kind of, often you'll see this as called the like insulation of the cell or the axon. And we'll talk about what that means before the end of class. But basically, the current flows, ions flow down through this insulated part of the axon, but actually turns out that the resistance is quite high. It is conducting, but the current that flows is slow. So that whole action potential, the ions that cause that have trouble actually flowing down the axon. Instead, what they do is that a little bit of current flows. And once enough charge builds up down here, it causes new ion channels in this region, in this space between the insulation, to fire and then build a new action potential there. And so it's like the action potential, these little nodes are called nodes of Ranvier. And basically they kind of rebuild the action potential. Since the current that flows down the axon is so slow, the new ion channels will fire when it sees a little bit of current flowing and then build a new action potential. And then that'll happen again and again and again at each different node and eventually take the signal down the neuron. So this process is called saltatory conduction. That's from the Latin or Italian verb saltare, meaning to jump. So that's your SAT or GRE or MCAT word of the day or whatever. I don't even know if they do have an English section on the MCAT. But so saltatory conduction means, saltare means to jump. So it's like the action potential jumps from one node to the other, and instead of kind of flowing in between. So it's actually, from a physics perspective, it's kind of cool. It's like you have this wave that kind of propagates down the axon. And so you have this wave in charges. The wave propagates from one node with ion channels to the next, and so forth. However, from the physics perspective, the unanswered question here is, there's a particular speed associated with this. It takes some time for the little bit of current that can flow to build up and cause these ion channels to fire. And that determines the overall speed of how quickly a nerve signal can propagate. And in that video I sent you from the Harvard Extension School, this is the one thing, they did talk about this in that video, but they never explained what determines that speed, just the fact that the action potential jumps, which is all good. But as scientists, I think part of our job is to have some understanding of why the speed is the way it is. It's really kind of a physics question. So what limits the speed of how quickly an action potential can move? That's the question I want to try to answer today with some of the physics that we've learned. So any questions at this point? All right, those of you may have, if you've had a physiology or taken neuroscience courses, you may have talked about some of this at some point. For those of you that haven't, that's kind of why I'm reviewing this. So hopefully this is clear. Again, as far as the exam goes, I'm not going to test you on any of the biology. It'll really be with the physics related to the problem, as usual. So here's the thing. So as we model our axon, let's zoom in on one of these little, or one or two of these nodes in the axon. So here we have the axon of our nerve cell, where my laser pointer is, or my virtual laser pointer, that's our cell membrane. And this big pink part here is the myelin sheath. And then we have two nodes. We've already talked about how the cell wall or the cell membrane acts like a capacitor. And the conducting fluid in the center of the cell is conducting, but it's not very great. So it's not really best modeled by a wire, but maybe by a resistor or a set of resistors. So if we wanted to draw like a simple cartoon almost, but like schematic circuit for what we're looking at, we have something like this. The ion channels that are firing and flipping the potential are acting like a battery in the circuit. They become a big source of potential energy or voltage, and that causes a small amount of current to move down this one node of the cell through this resistor. And at least as an approximation, we can assume that current's going to be given by Ohm's law. Obviously, things are not really that simple, but what we're really trying to do here is get a ballpark estimate. That charge eventually gets down over here and builds up on the cell membrane. So it kind of builds up charge on this capacitor. So really what we have here for this one or two node part of the axon is acting like an RC circuit or a resistor capacitor circuit. So what's really going to determine how long it takes for charge to build up down here and trigger the next axon or the next ion channels in the next node, that's really going to be determined by how long it takes charge to build up on this capacitor, which, as we learned about yesterday, was really given by the time constant of this RC circuit. So it's like after this capacitor charges up a small amount, we don't actually know how much it needs to charge. Does it have to charge 50% full, 75%, 90%, 100%? We don't really know right now, but after it hits a certain point, it will cause the next set of ion channels to fire and turn on the next battery. The process will then happen again until this capacitor charges, which turns on the next set of ion channels, turning on our next effective battery, and so on and so forth. So really what I'm going to, I guess, posit to you or claim is that the effective charging time of our cell wall or cell membrane capacitor is what determines how quickly this action potential can move. So we could really use the RC time constant as at least an estimate of that charging time. Now, if we go back to Wednesday's lecture that R times C, that time constant gave us the time it took for the capacitor to get 2/3 full. So I don't really know that that's when the next set of ion channels are going to fire, but it's going to be somewhere in that range. It might be 2 times RC, it might be 3 times RC, but this should give us at least a ballpark estimate of this travel time for the action potential. So we could actually get a bit of a measurement for the resistance between the nodes of an axon. I'm not going to worry about how we got that, but it's given in our textbook as around 25 mega ohms, so 25 times 10 to the 6 ohms. This is something that you can get current through something with this resistance, but it's really not a great conductor at all. It is conducting, but not really great. So it's something, so it's kind of a good model for the inside of our cell. As far as the capacitance of a cell membrane, the capacitance is determined by essentially the geometry. Though I didn't give you guys the formula for how to do it, it depends on the distance between the two plates and their effective area. So we kind of have the measures of those distances for the cell membrane. So using those, you can compute that the capacitance of the cell membrane is approximately around 1 or 2 picofarads. In particular, it's 1.6 picofarads. So 1.6 times 10 to the minus 12. So if we wanted to estimate the charging time between the firing of the different nodes, we could use our time constant, R times C. Sorry, there's a typo here. This 4 should not be there. So that's a typo. So R times C is about, we get about 40 microseconds. So this would be the time it takes the action potential to essentially build up enough charge here to fire the next ion channels. So this is the time it takes the action potential to jump from one node to the next. So this takes about 40 microseconds to move this distance. Let me erase what I just drew because I drew over. All right. And just by using some microscopy and by looking at these cells up close, we can estimate how long one of these nodes is. And about 1 millimeter. At least according to our textbook. I didn't take these measurements myself. So if we wanted to get an estimate of the speed of an action potential moving due to this RC circuit model, we just use distance divided by time. So that speed gives us around a speed of 25 meters per second. So this is the speed, for the action potential jumping from node to node. So this is effectively an estimate of the speed of saltatory conduction in a nerve axon, in a neuron axon. So whether this number is like reasonable or not, really what we want to do is, compare it to, we can do measurements using electrical measurements for measuring nerve signals that go from move down an arm or down a leg. So this is something we're actually able to measure in a lab. And so if we wanted to take this speed and think, so if I look at just pretend I'm actually measuring the distance from my head to my hand right about now. So it's, maybe if I hold my arm out straight, it's about 1 meter long. So as a rough estimate, the amount of time it should take a nerve signal to get, from my brain or from the, top of my spinal cord down to my fingers in my hand to hit the mouse to move to the next slide. That signal should take about, that total time for head to hand should be that distance divided by the speed we just found, which is about 40 milliseconds. So this 40 millisecond speed, it's about just right. It's not exactly the speed of the experimental speed of a saltatory conduction, but it's pretty close. So this is obviously a really rough model, but the useful thing about a rough model like this is that it tells us something about the fundamental scientific principles that are at play in the problem. That really the limiting factor on the speed of a nerve signal propagation is really determined by the resistance and capacitance of the nerve axon. And it's acting effectively like an RC circuit. Perhaps it's not just one resistor in there. Perhaps we have to account for the resistance of the extracellular medium, the stuff outside the cell. So we can make better and better circuit models, but just using the simple model from class, from our first year physics class, we can get a close estimate of this speed. So it tells us that really the nerve axon, the theory of resistor and capacitor circuits, is somehow at play in the properties of nerve signal movement and nerve signal propagation. So it's I'm not claiming that this is a like super accurate predictive model as built right now. But what I'm claiming is that it's telling us something that's, you know, true and hopefully interesting about what's going on electrically in a neuron. In particular, it helps explain something like, you know, this is a decent ballpark estimate, but this number explains, in particular, why this this simple model explains why this myelin coating is important to the cell. So, for instance, the reason this is there, you'll hear in physiology textbooks that really this is there to provide the insulation for the cell. And that is correct, but what that really means for our model is having this there effectively increases the time constant. Or effectively, having that insulation there will decrease the time constant. Because what it will do is overall decrease the resistance in the circuit outside the cell by having this insulation there. So this myelin changes our time constant, and it effectively allows the nerve signal to propagate faster. So some axons, some neurons do not have myelin coatings, and the nerve signals do propagate much more slowly. It takes a lot more time for this effective capacitor to charge up and the next node to fire. So the reason why nerve axons for motor movement have evolved to have this coating is to decrease the time constant of the effective RC circuit for the cell and increase the propagation speed. This also kind of explains why patients with multiple sclerosis have impaired motor abilities, because multiple sclerosis is an autoimmune disorder, for those of you that don't know, that where a person's immune system attacks the neuron cells and damages the myelin coating. And the damage can be, you know, just in part of a neuron, one part, but then everything on another part of the neuron could be fine. So you'll have these damaged nodes, which represented by these dashed black lines here, where effectively, due to that damage, the time constant is much higher. So it effectively disrupts the propagation of a nerve signal, of the action potential. Because you'll have nodes where the time constant is really short, the nerve signal propagates just fine, and then it hits this node where all of a sudden our capacitance and our resistance are thrown off by the damage to the myelin coating, which then slows down the time of firing for this next node, which gives you this kind of unpredictable but impaired motor ability and coordination in patients with this disease. Another place you see this is, for instance, you can see this model at play in giant squid. So for those of you, I don't know if those of you who have seen this or not, species of squid have nerves throughout their body that have evolved to have these gigantic axons, like close to 1 millimeter in diameter. So you could actually see them, this is like a dish where you can see them by eye easily. And this is not because the cell is 1 millimeter in diameter, it's because the myelin coating has evolved to be really, really thick. And what that does is allow for nerve signals to propagate super quickly, much quicker than they do, for instance, in our motor nervous system. And essentially the theory here is that they've evolved this to have a really fast escape response when they see some kind of threat. So they've evolved to have this really fast reaction time, really fast reflexes to help escape from threats. And they've done so by basically evolving really thick myelin coatings on their nerve axons to lower that time constant as much as possible. So if you guys have seen any of this in other classes or wondered why this myelin coating affects nerve signal propagation, it's all due to the physics of resistor and capacitor circuits, at least on a, at least on a basic level. in some way the circuit might be a little more complicated, but really it's just kind of, those are details. The general physics and scientific principles are all contained in this model that we're doing. So from here, and this is kind of the end of our material today, and kind of the end of our discussion on electricity and electrical circuits. And hopefully, I mean, I hope for a lot of you this problem was at least interesting, and I hope I answered some question that you may not have known before about, or I hope you learned something new here. If you talked about this in other classes, perhaps it was a review. But hopefully it was interesting for you guys and shows you where this physics we've been learning over the past few weeks, how it appears in nature. From here, the homework for today is going to be 4 problems. And more or less, most of them involve this type of problem that I talked to you guys through about predicting speed for action potentials and effectively speed for nerve signals to propagate through a body. You might be working through it in reverse or starting with some different numbers and such. But this problem here, from this slide to this slide, is a good model for the problems you're going to get in the homework today. And there are 4 of them. 3 mathematical and 1 conceptual problem. But that being said, that's the end of our material for today, and I think I'll end a little bit early, though I'll hang out if you have any questions. Otherwise, we're done for the day. Have a great weekend.